A very good evening to everyone. Let's crack CSIR-UGC NET together. The topic of this lecture is genetic drift, which is a part of evolution, and we'll be with it. So, before beginning with the topic of today, let us have a small introduction about myself and then Unacademy. I'm Farida Bandookwala, and I'm pursuing my PhD from TAVV School of Biotechnology. I've done my graduation, post-graduation and MPhil in biotechnology. I've qualified MPSTEC examination in year 2018 and I have a teaching experience of 10 years. So I told you we'll do a small introduction about Unacademy. So Unacademy is one of the India's largest platform for the preparation of NEET. What you will get at Unacademy is live classes, you will get courses, live quizzes, and unlimited access to all the videos available at the platform of Unacademy. And what is the benefit of daily live classes? You will always feel linked with the preparation. You will always have something to look upon, that yes, tomorrow this class is scheduled and this time this class is scheduled, so you will always be motivated towards it. Then there are structured courses. The courses are designed in such a way so that you can complete the whole syllabus in a limited time duration, because CSIR Life Science itself is made up of 13 units. This consists of 30 units. So it needs to be structured. So the course is designed in that way. And there are live tests and quizzes. So that is also possible with the Unacademy. And as I always say, whenever you want to qualify some kind of examination, it is very important to go for the tests and MCQs and PYQs. So that is also possible with Unacademy only. Then if you're going for plus subscription, you'll get unlimited access to all the recorded videos which were once part of live lectures. So you will get that also. Then Unacademy conducts free live classes on two platforms. One of the platform is YouTube. It conducts free classes on YouTube, and it conducts free classes on the special platform. These are the two places where free classes are conducted. So you can attend the class on YouTube and special class. You can ask your doubts in the live class. And YouTube class is just like what we are doing now. This is live. If you are listening live, it is live. And if you are listening after it is done, then it is in the form of recorded video. And then special classes, they are also free live classes. For this, you need to download the Unacademy Learning app from the Play Store. And when you have downloaded the app, you can make your account, you can log in, and you can have the access for all these special classes. So this was about the free. So maximum syllabus we try to cover in free and live classes also, and some of the syllabus which is not over here, it is in the plus subscription. So let us talk about this plus subscription. Plus subscription contains all the 13 units of CSIR Life Science in a well systematic manner, arranged manner, and each and every topic is covered systematically over there. And for this, you need to pay a very pocket friendly amount, which is not much. You can easily go for it. And if you're going for a 12 monthly or 24 monthly plan, it will be more cheaper to you, and it will also add to your benefit and add to your knowledge. If you are going for a longer duration, you can always look for a single topic by so many faculties, they have taught it. You can go for that. And lastly, you can use my referral code FB01 for getting a 10% discount. So do not forget to use my referral code, that is FB01. So the story of today is genetic drift. The story is of genetic drift. Now, genetic drift, what is it? We have already seen it. It's actually thing which is related to evolution. So genetic drift, before moving directly to its definition, I would like to make it in a very simple manner so that you can form the definition on your own. So genetic drift, where does it occur? The most important thing, where it occurs, to it is seen in small population. Um, Seen in a small population. If population is very small, genetic drift can be seen. Then it leads to random changes. If it is happening, it leads to random changes. Like it is not directional. No one has pre-decided that this is going to be selected. It is completely random. It is just a matter of chance. Random changes, then which are directionless, which are directionless, which are not having any kind of direction, which are just random, and it changes the frequency of allele. Changes frequency of allele. These are the three consequences because of genetic drift: leads to random changes, directionless, and changes in the frequency of allele, and it is seen in a small population. A very small population is going through genetic drift, then definitely it is going to encounter these things. Now, if you are curious that how I would write the definition or what is the definition of genetic drift, you can write it in a way that the random changes which are directionless and which leads to changes in the frequency of alleles and observed in a small population is called as genetic drift. So how would we write it? Random changes which are directionless leading to change in the allele frequency observed in small population is genetic drift. So what we wrote in the points that we have just written in a sentence form. So they should be random. There should be no selection pressure that only this will be favored, that will be favored. No. If it is a less privileged variation or less privileged allele or a diseased allele, then too, it's just a matter of chance. So random changes. There is no direction. It will lead to change in the frequency of the allele. Then it is called as genetic drift. So this is the thing. Now if we want to understand it by example, so give examples. I'll give the example. So how would you actually understand this? See, if this is an area of your garden and this is a forest nearby the city you live, and there are trees. So n number of trees are there in this forest. This is your garden and this is the forest nearby. So if there are 100 trees in your garden and in the forest there are 10,000 trees, and in your garden you cut 10 trees and in forest you cut 10 trees. So in your garden if there is 10 trees less, then obviously you will come to that change that yes, something is missing because it's a small area. And if in forest you cut 10 trees, you won't be able to demarcate because it's such a big large area and there are so many trees, you won't be able to observe the thing. So 100 trees and 1000 trees, so a small population. Genetic drift is observable in small population or it is seen in a small population. So in large population it won't be easily seen at all, right? So if 100 trees are there in the forest and 10 trees are gone, you cannot observe the change. And if there are if there are 100 trees and 10 are gone, you can observe the change. And if there are 1000 trees and 10 are gone, you cannot observe the change. So this is the concept of genetic drift, that how will genetic drift be seen. So genetic drift is seen in a small population. Always remember, genetic drift seen in small population. Never forget this, that it is seen in a small population. Now, types of genetic drift. What are the types of genetic drift? So genetic drift, they are basically of two types: bottleneck effect and it is founder effect. Bottleneck effect and founder effect. Two types of genetic drift are seen. So both bottleneck effect and founder effect, they are similar in their consequences, but the reason why bottleneck effect is happening and why founder effect is happening, the reason might be different. So they are similar in their consequences but different in their reason. Similarity is more, differences are less in them. So they both are having high similarity and at some places they are different. So result of both the effects will be same. Result of both the effects are same. Right. So bottleneck effect we are going to deal first. Bottleneck effect. So I would like to explain this bottleneck effect with a picture. Yeah. So this is the picture expressing bottleneck effect, genetic bottleneck effect. Can you observe the bottles over here and glasses over here? This is a bottle. How many colored beads you can observe in this? You can see white, you can see green, you can see yellow, you can see red. So these are the four prominent colors which you can see in the bottle. Now, if the bottle is having these number of beads and the bottle is inverted in a glass, so the opening of bottle is quite small. So all of the beads, they won't go down altogether and there will be a blockage in the movement of the beads in the glass. So can you see the glass in the second place here? So can you see that only blue and yellow color beads have come in the glass? And when they were tilted for a longer duration, it was just a matter of chance that only blue and yellow beads came in the glass. Rest all red, green and white, they were left out in the bottle itself. So in the bottle, red, green and white along with blue and yellow are there. And in the glass, there are blue and yellow only. So this is what actually tells us about is that bottle has large number of population variation. The bottle has got population variation. Right, large number of varieties. If you talk in terms of alleles, large number of alleles are there. You talk about a single species, so if in this bottle a single species is there and they are carrying so many different types of alleles, but when the bottle is tilted, only some types of alleles they come in the glass, not all the alleles. So this bottle is our environment. And the alleles which are moving into this class is because of some other factors. That the other factors can be many, so they can be because of the many changes, right? So that I'll be just telling you. So the glass has only a small population of the survivors, that is only two colors, means the variations are lesser, variation has reduced. Population variation is high here in bottle and here variation is less. Variation reduced, it became lesser. So the surviving population is having lesser variation. So coming further, that on seeing the two colored balls, we can say that the other colored balls were also existing from this this glass. Can you guess that the original bottle also contained red, green and white colored beads? No, you cannot guess out from this glass. From this glass, you cannot guess out from where it has come from. So, from the surviving population, one cannot understand, one can't get that the original population had other alleles too. So from the surviving population, one cannot get that the original population had other alleles too. What another thing you can get from this, that there were lots of variation in the bottle, but in glass only two types of alleles are there. So variation is less in the surviving population. So variation is less in surviving population. Variation is quite less in the population which actually survive. These are the things which you can understand from the bottleneck, that the surviving population, it is having only two alleles by the color. Just a hypothetical explanation I am giving based on the color. If colors they are the alleles, so just two alleles are there. And in the original population there were so many alleles. So you cannot guess from the beads which are in the glass that originally there were red, white and green also. So that is the thing you can make out from the bottleneck effect. Now the most important thing which I was telling you, that I'll tell you the reason of bottleneck effect. So bottleneck effect, reason of bottleneck effect. Reason of bottleneck effect. So why is it called as bottleneck effect? The mouth of the bottle is very narrow, and whenever you are putting some colored beads like thing in it and you just try to invert the bottle, some of the things will randomly come out. So that is just related to the concept bottleneck effect. The explanation, the pictorial explanation or the name, what it would mean, and it will cause an impression in your mind. That is why the term bottleneck effect is used. So most important thing, what are the reasons of bottleneck effect, isn't it? We definitely need to know that what are the reasons of bottleneck effect. So it can be random death, a random death of the population, maybe because of a disease or something, the random death of the of certain people in the region, like maybe because of a volcanic eruption, the people they died because they were just near to that thing. So death or natural disaster will leading to death. Natural disaster, which is leading to death, like natural disaster can be flood or earthquake or volcanic eruption. So anything can lead to death and natural disaster. Otherwise, death, random death. It should not be selectively on the basis of the genes. It should be a random death. That if a person having a stronger genetic makeup or stronger immune makeup is also coming in that environment where it will lead to death, that person will also suffer death. So death, natural disaster, then hunting, random hunting can also lead to bottleneck effect. Imagine all the tigers, they are hunted for the poaching purpose and just some of them are left out. So there is lot of variation, and if just a small number are left out, for example, if there are 1,000 tigers all over India and all of them are hunted and now just only 10 are left or 20 are left, so the genetic variability in the 20, that will only be propagated further. or it will be passed on further. Rest, all the variations would be eliminated out from the population. That is the hunting. So there are many things, many examples which are even based on hunting, which are related to bottleneck effect. Then fragmentation of habitat. Fragmentation of habitat. The habitat would be fragmented because of some natural things, some drifting, some geographical location. It is getting divided into two if a mountain appears or something like that. So fragmentation of habitat is also the reason for bottleneck effect. Now we'll be seeing some of the examples for bottleneck effect. Yeah. So the examples of bottleneck effect. Bottleneck. So what is the very first example? You can see that by 1900, hunting of the northern elephant seal of the Pacific coast had reduced their population to 20 survivors. So there were many, but they were reduced to 20 survivors because of their hunting. Since hunting ended, the population has rebounded from the population bottleneck to some 100,000. So in 1900, when the population was reduced to 20 and when a check was made on their hunting that now they cannot be hunted, they again took their natural size in that habitat. So they again became 100,000. So now today when there are 100,000, all of these animals are homozygous at every one of the gene loci they have been examined. So homozygosity exists at each and every gene loci. It means the variation which was there in their large population is just wiped out, because from 1900s the time elapsed is quite less. So they haven't got enough time to develop the variation also. So the immediate effect of bottleneck is reduced number of the allelic gene frequency. The gene frequency has just come down so much. So this is a very important example of bottleneck effect. The first example is of northern elephant seal. There is a southern elephant seal also, and the southern elephant seals, when the northern elephant seals are compared with the southern elephant seals, what is observed is that in southern elephant seals there is lots of variation still present. Still they have got large number of variation. So as compared to northern elephant seals where there was lots of hunting, their gene frequency has come down. Now the second example is about cheetahs. Cheetahs have passed through a similar period of small population size. Its accompanying genetic drift. So cheetahs, they also had to face this unfortunate condition. The examination of 52 different gene loci was done and has failed to reveal any polymorphism. They were completely homozygous for those 52 loci. That is, the animals were homozygous at 52 loci. The lack of genetic variability is so profound that cheetahs will accept the skin grafts from each other just as identical twins, and inbred mouse strains too. So if there is lots of variation, there will be difference in MHCs, there will be lots of immunological reaction, but they were inbred because of the lesser number of them. They can accept the graft of others. So the thing which we want to actually convey by acceptance of graft from anyone, what we want to say is they are so similar from each other with each other that there is no rejection in grafts, and they are similar at 52 different loci. And for elephant seal, they were like totally similar at all of the gene loci. So this is the example of genetic drift. So these are two of the examples which I wanted to highlight. Now other example also which I want to highlight over here is about antibiotics. So this is a hypothetical example. These two are the actual impact of bottleneck in the recent time which we have actually studied on. So northern elephant seal and cheetahs, do not forget that they have suffered bottleneck in recent past. Now talking about the bacteria, now what happens is whenever a person he suffers from some kind of bacterial infection and he goes to doctors they prescribe antibiotics, right? So what happens is the bacteria will be wiped out, which are actually pathogenic to humans, by the usage of this antibiotic, right? So those people who are consuming antibiotic regularly, the dose which has been prescribed, those bacteria will be completely gone out from the population or gone out from there. But those people who would skip out the antibiotics, the people who are not consuming antibiotic properly in that dosage, their bacteria will survive in that environment. So that means their genetic variability is reducing, but yes, some are surviving because of the chance. Humans, they are not thinking about bacteria, but they are just thinking about themselves, and they are just skipping the antibiotics. So that will lead to chance survival of bacteria. So that is not any kind of added advantage that they are developing some drug resistance or something like that. No, the person who is actually having the disease himself thinks that no, I don't want it. So this is a chance survival. So these are some of the very interesting and important examples of bottleneck which you should not forget at all. Right now, just a second. So yeah, so two examples we have seen, and we have seen this. Now we will be talking about the founders effect. Yeah, so this is the picture of founders effect. So this is the ancestral or original population. Here you can see this is the ancestral and original population, and this is the founder population. Now what happens is, from a large area, if a certain group of people, they think that we would like to migrate from here. No specific reason of migration, just they want to migrate. There is no specific reason that for this reason we are migrating. They are just migrating like that. They are migrating like that without any reason. So can you see that some people, they will form a group and they will migrate. So when they will migrate in this new habitat or new place, they will have limited number for their reproduction. They will have limited number for mating. So they will mate in own, and because of their inbred mating, some of the genes will, which were actually in lesser frequency in the previous population, they will increase in the further population. So this can also be seen from these two charts. See, the ancestral population frequency for red and green in the original, initially it was equal, like for red and green. So here they are appearing, yeah, red only. So red and green, so initially red and green, they were just equal in number. Then after the split, red became a little higher than the green in the generation. For the ancestral population we are talking, red became a little higher, and then in the further generation still the changes were not that profound. Okay, fine, they were around 0.57, 0.43, so they were like quite close only. So in the ancestral population or the original population, there were no major changes or major differences it caused because of this migration or moving out from that particular position. And in the founder population, which actually moved to some other place, there were major changes which were seen. So the major changes, initially the red and the green, after the split, the red were quite less and green were quite high in the original population. In the generation one, red improved in number, green went down, because some of them, they were having this green allele in them. It was not like that they are not having at all, because they are having in their genes, but it is not exhibiting. So here you can see just red colored dots. So because it is not exhibited in the phenotype. Then in generation one, again, when it did not get more of the other green allele, it became red one became higher and green one became lesser. And till generation 5, they were all completely made up of the red allele, and green allele was completely wiped out. So this is the founders effect. What happens in this? So founders effect, we have seen everything about founders effect, but we haven't seen the reason of founder effect, why the founder effect actually occurs. Like in the bottleneck effect, we have seen that why does bottleneck effect occur. So we need to know the reason of founder effect, right? Reason of founder effect. The most important thing whenever we are doing anything, we need to know the reason, right? So the reason of founder is founder effect is no specific reason to migrate. So they are not having any specific reason for migration, but they are migrating. Thing which is happening to them is migration. The reason of founders effect is migration, but no specific reason of migration. Migration is occurring, fine. Okay, they are migrating, but there is no specific reason why they are migrating. This is the thing which is associated with the founders effect, that there is no further reason of migration, but they are migrating. So the main role which is played in founders effect, so you should just write in your mind, most importantly, and the copy which you are using, migration. So you should not forget FM, founders effect, migration. FM, F for founder and M for migration. So do not forget FM. Founders effect is because of migration. So original population, initially there were some of green, more of green and yellow. They were almost green and red. They were almost same in number. Then when they migrated, initially there was a stage when they were having red and green, both of them. So there was a time where when it had red and green also. But with time, when red were dominant and red were more, red became more profound in the coming generation. So it is established and forms the colony. These red, they establish themselves and make the colony, and it will lead to founder population. This is called as founder population. Founder population. And they will face a selection pressure, and they will also interbreed because of the why the red is becoming more and more, because of selective pressure. They will have to faces selective pressure and inbreeding, because they are not getting lots of people to mate with, so they will undergo inbreeding. So now after large number of time, only some of the alleles are uh, left out or flourishes in that environment, and rest of them are wiped out. So founder population will never now, some after some time, just as this. Now what is the consequence? So like we did this bottleneck effect, we also read that is it possible to identify the original population from the um, population which has emerged from the original population? So the answer was no. We cannot find out who all were there in the bottle. We just know what is there in the glass. The thing which is there in the glass cannot tell you what was there in the bottle. So here also the founder population do not give us any idea about the ancestral population. So we can write one sentence over here importantly, that founder population do not give idea, do not give any idea about parental population or ancestral population. Ancestral population. This is the thing. They do not give any kind of hint. And obviously you can see here the variation is large because there is red color and green colored allele, and here only red color allele is there. So the most important thing is variation is lesser. Variation is lesser in founders effect too. Right. So the consequence, can you see the consequences are same? The consequences of bottleneck effect and founder effect, they are same. Just the reason is different. So we can write one sentence. Yes. So bottleneck effect and founders effect. Consequences are same. Consequences are same. Reasons are different. Reasons are different. Consequences are same and reasons are different. Now, when we are talking about bottleneck effect and we are talking about founders effect, we have seen beautiful examples of bottleneck effect. That is the northern elephant seal, and we have seen about cheetahs. Then we have seen about antibiotic resistance in bacteria, and not antibiotic resistance, but how the bacteria, they are wiped out by antibiotics. Now let us have a small discussion on the examples of founder effect also. So this is one of the example of founder effect. See here, the result of genetic drift, especially I should write over here founder effect. Founder effect. So the founder effect, it is seen in the Amish babies. So there was a ancestral population. From them, some group of people, they actually migrated to some other place, and there they had to suffer from polydactyly. This situation is polydactyly, having more than five fingers, 5 digits. So they had to suffer polydactyly. So this is not because of any kind of mutation or any kind of deformity in them, but the original population had this allele of polydactyly, but they were so many in number, it was not profound in them, or we cannot just generalize that all are having polydactyly. But when a group of people, they migrated by chance to some other place, the major gene which became profound in them was of polydactyly. So the founder effect in action is among the Amish babies with Ellis Van Creveld syndrome are born with 6 fingers. So there is one Bollywood celebrity also having 6 fingers. We all know him very well. That is Hrithik Roshan. So don't relate it at all with him. He doesn't have this thing. So, but still, just for making you remember that polydactyly is seen in the Amish babies, and Amish babies had this polydactyly because of founders effect. They were actually migrated from some other place, and this gene became homozygous in them. So almost all of them were showing this trait. Then another example of founder effect that we need to see is of porphyria, mild metabolic porphyrias, which are related to the heme metabolism. Porphyria. So in 1680s, there were two people who migrated from Holland to South Africa, and one of them, they were having allele for the mild metabolic disease, that is porphyria, which is related to heme metabolism. So today, more than 30,000 South Africans, they carry this allele, and it is said that this allele is dating back to that couple, to this couple, a remarkable effect of founders effect. So this is a very, very interesting example of founder effect, that how a population of 30,000 dates back to just a single couple which came from Holland to Africa carrying this allele, and now this allele has become so profound in the South African people that they are suffering from porphyrias, that is a metabolic disease. Then I have included one more example of founder effect so that you cannot forget at all founder effect. So this is about Huntington's disease. Huntington's disease. So it is about the Dutch who colonized South Africa, and this is also dating back to the same era when where we have seen this porphyrias. So few colonists carried relatively high frequency of allele, which were actually responsible for Huntington's disease. So Huntington disease, it became more prevalent amongst the Africans, and then as compared to other people in the world. So this is a classical example of Huntington's disease, in which people, they suffer lots of um, kind of, can say, dementia or amentia. They actually suffer in Huntington's disease. So founder effects are easy to recognize for genetic diseases, but of course founder effects affect all sort of genes. So here we are seeing some of the genetic diseases, but all of the genes, they are affected by the founder effects. And founder effects, we have seen some of the major examples which are related to humans in founders effect, and when we receive seeing bottleneck effect, mostly we have seen the example of animals. So do not forget to remember these examples because directly if these examples are quoted in the exam, you won't have to think much about it, and also remember the reason. Lastly, to conclude the lecture of today, we will be we'll be discussing certain points. So first is the bottleneck effect and founder effect. Both are kind of genetic drift. Both are kind of genetic drift. Then bottleneck effect and founder effect. Commonly, if you are saying genetic drift, they are responsible for explaining various evolutionary process. Can explain evolutionary process. Can explain evolutionary process. And if we are saying evolutionary process, so specifically they can explain about speciation. Can explain about and what is other thing it tell? It can also tell about fixing of harmful genes. Fixing of harmful oblique lethal allele. So we have seen in the founders effect lot of the things which are actually uh, the effect of fixing of harmful allele. The Amish babies, the example of Amish babies, the examples of porphyria, the example of Huntington's disease. These are the lethal alleles. So fixing of harmful allele is example of the founder's effect or genetic drift commonly. So random event which is significant in a small population can lead to genetic defect drift. So genetic drift occurs in a small population, occurs in a small population, and this is small population with a random event. In a small population due to random event, there is no specific reason for any kind of thing which is occurring, so it is due to the random event. And what else is it causing? So it operating on the basis of genotype. Mostly what you have seen is the expression in the phenotype in founder's effect, but deep within it is because of the genotype. So operational basis is genotype. It is the genotype because of what this is happening. Then variation is lost in the new population. Variation is lost in, we have seen that in the bottleneck effect also there were 5-6 type of beads in the bottle. That means 5-6 types of allele for a single type of gene was there in the bottle, but when it suffered bottleneck effect, only some of the alleles came in the glass. And when we are talking about genetic drift also, only some of the alleles were fixed in the founder population. So variation is lost in the new population. And what else occurs in this? The surviving population cannot tell you anything about the original population. Very important thing. Surviving population is not going to tell you anything about the original population. It is not going to hint you at all that what all alleles were present in the original population. Right. So two of the things are still left. That is the reason of bottleneck effect is mostly natural disaster, and the reason of founder's effect is migration. Is migration. So So this is how we have completed this topic of genetic drift with detailed examples and whole explanation in a well systematic manner. So just a very quick revision for those who are attending the class from mid. So genetic drift is seen in small population. Do not forget that it occurs in small population. It is completely matter of chance. It is random. Then it is directionless. There is no specific direction in which it is going to move, and there is change in the frequency of allele. There will be changes in the frequency of allele. The frequency of alleles is going to change. So after reading the whole chapter, we are understanding this thing quite well, that how the change in the frequency of allele is going to lead to genetic drift. Then we have seen that how it is observed in a small population and cannot be observed in larger population, because once we have seen the example of a forest having 10,000 trees and a small garden of our house having 100 trees. Hi Himanshu Kumar, good evening, and hi Sid Nayak, hello. So you have joined the class from mid, so no issues. I am taking the revision part, so you can just see, and if you have any query, you can ask. So we were talking about the genetic drift, and it is seen in small population. It is a very random matter of fact, and it is directionless. It is not moving in a particular direction, right? So a forest having 10,000 trees, if 10 trees are cut down, you won't observe any difference in the whole genetic makeup of the forest, or by visual appearance also it won't be any kind of different. But when you are talking about a garden of your area, if suddenly from 100 trees 10 trees are cut down, you will be able to observe something. So this is just about the visual thing I am telling you about, but it will be highly impacting the genetic basis also. Now, the genetic drift are of two types, that is bottleneck effect and that is the founder effect. So bottleneck effect and founder effect, they are similar, and they are similar at most of the things, just they are different on the reason why it is occurring. So bottleneck effect and founder effect, they are just different why they are occurring. I hope you are understanding, Himanshu and Sid Natak. Now, bottleneck effect, the reason of bottleneck effect is because of the death, because of the natural disaster, because of hunting, and because of the fragmentation of habitat. So all these things, they are just very random. They are not specific. Natural disaster won't see that I need to affect this area. Randomly it will happen. Hunters and predators, they are not going to hunt and do it selectively. They are going to do it in a very random manner. Okay, if you are understanding, good. Great, I've seen your comment. Then death, if death is because of volcanic eruption or harmful gases, it will be very random. It is not going to be because of specific thing. So this is the thing about the bottleneck effect. Now this is the picture which is explaining you about the bottleneck effect. See, this bottle is having 5 colored beads, that is white, red, green, yellow and blue. But when this bottle is tilted in the glass, only 2 colored beads are there in the glass. So these two colored beads are telling you that now only these 2 colored beads are going to form the new population. And this bottle, maybe it is made up of 5 colored beads, it doesn't matter. But the thing which is coming in the class is only blue and yellow. So why it is explained by the name of bottleneck effect? Because the mouth of bottle is very narrow, and this narrow neck actually explains the whole concept. So that's why it is given the name of bottleneck effect. In any bottle, if you are putting some beads and you are just inverting it in the glass, you won't be getting all the beads in a very systematic manner. Okay, all the 5 color will come in the one go. In the one go, only some 2 or 3 colors might come and rest of won't come. So in the one go, whatever colors are coming in the glass, they'll be making the further population. So from the surviving population, you cannot at all understand. Can you make out from this glass that what actually original population was made up of? Can you see the glass and can you tell you that? Can you tell me that original it was also made up of white, green, and red? No, not at all. You cannot get any hint about the original population. And variation is becoming lesser. Obviously, initially it was having 5 colored beads. Now it is having just 2 colored beads. So here the beads, they are directly related with what? Related to the alleles of a particular gene, that now the gene is having lesser number of alleles in them. So variation is less in the surviving population. Then we have seen the founders effect, and founders effect is because of the migration. The random migration. They are not migrating on basis of lack of food or lack of anything. They are just randomly migrating. Like somehow people, they decide, okay, we want to just settle in a particular area. So like that, random. So in between this larger circle and smaller circle, there is a stage also where there were red and green color beads also, but after some time they became completely red. So from this table you can easily make out that initially red and green were there, but red, it became it dominated the whole thing. So why did it dominated the whole thing? Because of selective pressure it has to face, and the inbreeding. Because if there is only 5 to 7 people in a small group, just for example, I'm telling, if they are migrating, they have to inbreed, they have to inmate. So that inmating lead to increase in a particular kind of allele. So reason of founder effect is no specific reason of migration. It is migration, but there is no specific reason of migration. So just remember FM, the radio channels, FM. Founders effect is due to migration. FM, migration is the only reason for founders effect. Remember this thing. Now, founder effect, if it is occurring, can you make out from the circle that initially they were also having green colored allele? No, not at all. Can you make out? Yeah, selective pressure means. Yeah, selective pressure. I've written over here selective pressure and inbreeding. So what is happening? Can you make out from this smaller circle that initially it was having green and pink, red beads also? No, not at all. You cannot get any idea of the initial population, and variation became lesser and lesser in the upcoming population. Now, I have written some of the examples. We are dealing in a random manner because initially I dealt in a systematic manner. So, example of polydactyly is because of the founder effect. It is seen in the Amish babies. Some group of people, they migrated, and the gene for polydactyly, it increased in the founder effect. Then there is this example of founder effect for the porphyrias. They are related to heme metabolism. So a couple dates back to the 1680s, and they are responsible for 30,000 people having porphyria in today's time as well. Then another example of founder effect is Huntington disease. This also dates back to 1600s and 1700s. So it is kind of dementia kind of defect, Huntington's defect. Then bottleneck effect, it is seen in the elephant seals, northern elephant seals, and because of their hunting, just 20 survivors were there, and when there was a check on their hunting, those 20 survivors had to build up the population. So they took their normal size in that particular habitat, they increased in number, but the most important twist was that all the gene loci, they became homozygous for all the genes. Homozygous at all the gene loci, and same problem cheetahs they had to face because of this hunting. When they were left out and when there was a ban on their hunting, just now from all the gene loci, 52 gene loci, when they were seen, they were homozygous for those 52 loci. They were homozygous at that particular position. So this is the reason, this is the example of bottleneck effect. Now lastly, bottleneck effect and founders effect, the consequences are same, but the reasons of both of the things are different. And lastly, just to sum up the whole thing, bottleneck effect and founder effect both are part of genetic drift. Then genetic drift can give you a very good idea about speciation through evolutionary process. And and in founders effect, we have seen fixing of some bad genes, harmful genes like Huntington's, Amish babies, and porphyria. So it can tell you a very better in a way about the lethal alleles also. Then genetic drift occurs in a very small population. It does not occur in a large population. It occurs in a small population, and it's a very random event. It is operated in genotype. Like, okay, we have seen it in the phenotype like Amish babies and Huntington's and porphyrias. We have seen in the phenotype, but basically it occurs in genotype, isn't it? So variation is lost in the new population and surviving population. By saying surviving population, you cannot tell that what existed in the original population. You will not get any idea about the original population. And reason of bottleneck effect is because of natural disaster, and reason of founders effect is migration. So like this, we have done this topic in a very interesting and very detailed manner. If you are understanding this topic here, you will be easy, it will be easy for you to make the questions out of these, and we'll be doing a question answer session also on this. But for now, we have done the theory. we'll be talking about molecular clock and Hardy-Weinberg principle. You know how much important it is, exam point of view. So just be there in the class. Sid Nayak is saying, thank you so much, ma'am. Can you give your email address for doubt? Sid, you can, if you want to message me personally, what you can do is you can go on Unacademy Learning app, make your profile over there, and then follow me. Search me by my name, that is Farida Bandookwala, and there is an option where you can message me personally. So there we can have all the doubts cleared in a systematic manner. I hope you are understanding. Just go there and you can get me over there and search me by name Farida Bandookwala or Farida Bandookwala. Easily we can talk over there in detail. That is a personal chat.